Morning, students. Today, in continuation with chapter Biomolecules, we shall be studying about the structure of protein. Protein are the polymers of alpha amino acid. They are connected to each other by peptide bond or peptide linkage. Peptide linkage is an amide linkage formed between COOH group of one amino acid molecule and NH2 group of another amino acid molecule by the elimination of water molecule. Dipeptide is made up of two amino acids. For example, when carboxyl group of one amino acid combine with the amino group of another amino acid, we get the dipeptide. In dipeptide, there are two amino acids molecule. A dipeptide consists of two amino acids. If third amino acid combines to a dipeptide, the product is called the tripeptide. It means the tripeptide contains three amino acids linked by two peptide linkages. Similarly, when 4, 5 or 6 amino acids are linked, the respective products are called tetrapeptide, pentapeptide and hexapeptide respectively. A polypeptide with more than 100 of amino acid residue having molecular mass higher than 1000 U is called protein. And a polypeptide is formed when the number of amino acids are more than 10. So when more than 10 amino acid molecule combine, a polypeptide is formed. Next is structure of protein. It can be studied at four different levels, that is primary, secondary, tertiary and quaternary protein. First is primary structure of protein. Primary structure of protein, we can say that protein may have one or more polypeptide chains and each polypeptide protein has amino acid linked with each other in a specific sequence. This specific sequencing of amino acid is said to be the primary structure of protein. Secondary structure of protein, it refers to the shape in which long polypeptide chains can exist. It exists in two forms. One is alpha helix and second is beta pleated structure. The structure arise due to the regular folding of the backbone of the polypeptide chain due to the hydrogen bonding between CO and NH group of the peptide bond. Alpha helix. Alpha helix is one of the most common ways in which a polypeptide chain forms all possible hydrogen bonds by twisting into a right-handed screw with NH group of each amino acid residue hydrogen bonded to CO of the adjacent turn of the helix. Beta pleated structures. The beta pleated structures consist of polypeptide chains which are stretched out to nearly a maximum extension and then laid side by side, which are held together by intermolecular hydrogen bonds. The structure resembles the pleated folds of drapery and therefore it is known as the beta pleated sheets. Tertiary structure of protein. It represents the overall folding of the polypeptide chains, that is, further folding of the secondary structure. It gives rise to two major molecular shapes, that is fibrous and globular. The main force which stabilize the secondary and the tertiary structure of protein are hydrogen bond, disulfide linkage, van der Waals forces and electrostatic force of attraction. Then is quaternary structure of protein. The proteins are composed of two or more polypeptide chains referred to as subunit. The spatial, that is the arrangement in the space of these subunits with respect to each other, is known as the quaternary structure of protein. Now we'll see that the classification on the basis of the molecular shape. The protein can be classified on the basis of molecular shape into two. One is fibrous protein and second is globular protein. In fibrous protein, the polypeptide chains run parallel and held together by hydrogen or disulfide bond, and then fiber-like structure is formed. Such proteins are generally insoluble in water. Some common examples are keratin, present in hair, wool or silk, and myosin, which is present in the muscles. Then is globular protein. The globular protein results when the chain of polypeptide coiled around to give a spherical shape. They are usually soluble in water. Example is insulin and albumin are the common example of the globular protein. Denaturation of protein, again it's a very important topic. What is the meaning of denaturation of protein? Denaturation of protein means when a protein in its native form is subjected to the physical change like change in pH or a chemical change like heating, the hydrogen bonds are disturbed. Due to this, the globule unfold and helix get uncoiled and protein loses its biological activity. During denaturation, the secondary and the tertiary structures are destroyed, but primary structure remain intact. Example is curdling of milk and coagulation of white on boiling. It means that protein on denaturation, it loses its biological activity. Thank you.